Now, we will discuss each complex component of the liver. When we study the liver at tissue level, the microscopic view of liver shows the cells of the liver. The major cells that form almost 80% of the liver are the hepatocytes, and all the metabolic activities are done by the hepatocytes. Then we have our stellar cells, which stores fat and respond to injury. Then we have our Kupffer cells, which are the first innate immunity cells that gives protection of the liver. And the last cell are liver endothelial cells that helps to exchange the nutrients and blood flow. To memorize these 4 types of cells, you can use a mnemonic: hungry snake killed laborer. As we know, we have 4 cells of the liver, but the major portion of liver consists of hepatocytes, so they are definitely of prime importance. Let's study some more details about them. So, hepatocytes are basically polyhedral cells, which means they have 6 surfaces. They make up 80% of total liver portion and comprise of 2 to 4 nuclei in each cell. They have the ability to detoxify harmful drugs and chemicals in liver and also excess cholesterol. In addition, they also have the ability to store glucose in its inactivated form, which is known as glycogen. The hepatocytes also play a major role in metabolizing drugs and harmful chemicals, and it also undergoes triglyceride or fatty acid metabolism. Here we have an image in which you can see the hepatocytes. So now we are going to dive deep to learn the histological components of the liver. Right here we have an image in which we will study the 2 major divisions. The first division is the stroma of the liver, which is derived from a Greek word meaning bed covering, and it has layers of connective tissues, which are composed of reticulin, the type 3 collagen, and has a structural role. It contains all the blood vessels, nerves, and ducts. The other one is the parenchyma, which comprises of all tissues conducting specific functions, and they are mainly represented by a major portion of hepatocytes and other cells, as we have studied before. To remember these 2 major divisions of liver, we can use a mnemonic SP. Now we will learn the details of these histological components. The liver stroma consists of all the connective tissues, and stroma means layers or coverings. So the connective tissues of stroma are arranged in 2 layers. The first one we have is the serous layer containing the visceral peritoneum, which lines the liver and gives some sort of protection. The other one is the fibrous layer containing the Glissons capsule, and this capsule basically forms the outer protective coating of the liver lobules. Just like the capsule we take in our medications has a hard covering, in the same way Glisson capsule also act as a sheath to protect the liver lobes. In this image, we can see fibrous layer Glisson's capsule, and here we have the visceral peritoneum of the serous layer. Then we have sinusoids, which are the capillaries traveling between the hepatocytes, and it helps in cleaning the blood from the old red blood cells and other debris, and add nutrients in the blood so that it can be stored. Right here, we have a microscopic image in which a central vein can be seen, and here we have our sinusoids, which are networked in a star-like manner. Now, as we have learned about the protective capsule, the networking channels, and many other things, we are going to focus on some spaces inside the liver, which are spaces of Disse, also called as perisinusoidal spaces, which act as channels and allow the plasma components and proteins to move from sinusoids into the hepatocytes where they are absorbed. To get a clear understanding of where they are located, we can see in this image that we have our hepatocytes on the one end and sinusoids on the other end, and in between these 2, we have our perisinusoidal spaces. Right here, we have our parenchyma as described before. It is mostly comprised of hepatocytes, and it contains other cells too, which are the stellar cells, the Kupffer cells, and the endothelial cells, which we have learned before. This is a simplest illustration taken from the liver and visualized in a microscopic manner to show the location of the parenchyma. Parenchyma also contains some major structural components that are very important in liver functioning. The parenchyma contains the hepatic lobules, which consists of plates of hepatocytes stacked upon one another. So, talking more about the classic or hepatic lobules. These consist of hexagonal plates, and in this image of lobule, we can see that we have a central vein which is present in the center of hepatic lobule, and in the sides, we have a huge network of hepatocytes. Okay, so moving ahead, we have a triad consisting of 3 networking connective tissues called as portal triad, which is formed by combination of 3 central veins. We have 3 divisions of connective tissues associated with it. The first one is the hepatic artery that carries oxygenated blood to hepatocytes. The next one is the portal vein, which carries the deoxygenated blood. And the last one is the interlobular bile duct that carries the bile products away from hepatocytes towards the gallbladder. Just as the cells are the functional unit of body, the liver acinus is the functional unit of the liver, which is associated with blood supply. It is divided into 3 zones on the basis of gradient of blood supply. Here we have an image in which we can see liver acinus located centrally. A cone-like structure contains the 3 zones, as highlighted here. Now we are going to study each zone of liver acinus. The zone 1, also called as periportal or peripheral zone, because it is away from the center and is located closely to the adjacent lobules, and so hepatocytes receive the highest amount of oxygen. Then we have the intermediate zone or transitional zone, which you can visualize in this illustration. Zone 2 is in which the hepatocytes receive intermediate amount of oxygen. Now we are going to learn the last zone, which is the zone 3, also known as centrilobular zone, or the central zone located away from the periphery, or the shorter axis, but is located closely to the central vein, and hence the hepatocytes receive the least amount of oxygen. So we are almost done with the details, but right here we have a structure called as biliary tree, which is a system of channels that carries bile and help to store, secrete, and transport bile. In the image of human liver, the one illustrated in green color is the pear-shaped organ gallbladder, from where we can see the biliary tree originating along with its branches. The smallest division of biliary tree is the biliary canaliculi, which are the smallest channels, and right here we have our central vein, as we have seen it multiple times. Now in between all the hepatocytes channels, we have biliary canaliculi. Hepatocytes secretes bile into the biliary canaliculi, which then forms the loop of canals of Herring, from where the bile moves into interlobular bile duct, which we have studied previously in portal triad. Making our way towards the end, let's take a look at the vascular supply of the liver. We can see that the major portion comprises of hepatic portal vein, contributing 75% in vasculature of liver, which carries deoxygenated blood from the spleen, pancreas, and the intestines. Then we have the hepatic artery, which contains the oxygenated blood and contribute 25% in vasculature and brings oxygenated blood to liver. So that was all for today. Stay tuned for more histology medical videos. You can even search our library for other topics that you want to study, only on sqadia.com.